Imagine you have been charged with building a robot. What are the most advantageous characteristics to build into such a machine? To perform work, energy will be needed. Renewable energy extracted from the environment is ideal. To respond with meaningful behavior, information gathering and processing capabilities will be required. For coordinated operations, communication with other robots is essential. To maintain a long-term function, a reproduction system will be necessary. While all these requirements are significant hurdles to the robotics engineer on a budget, they are feats that life has accomplished time and time again. Consider bacteria. They are only a few micrometers in length and capable of all of those things. An engineer building a device on a bacterial chassis would only need to build one functioning prototype, culture overnight in low-cost media, and return the next morning to obtain trillions of virtually identical copies.
What functions are we capable of programming into a living organism? What is the best way to implement a predefined design goal, and what challenges and opportunities may arise?
Over the past few years, synthetic biologists have generated remarkable systems of E. coli, rewired yeast mating, and created genetic switches in bacteria.
The laws of physics and chemistry apply to non-living things and living things alike. Whereas mechanical engines can be engineered predictably, simple biological systems are a challenge.
Synthetic biologists, therefore, aim not only to produce interesting and useful designs, but also to simultaneously develop a greater understanding of biological components and design principles in general.
It's important to gain biological insight from testing our knowledge during the design and implementation process.
Just as the microscope made the discovery of cells possible, new technologies are providing the critical foundation for synthetic biology, such as DNA synthesis, which allows us to reprogram an organism. Until recently, DNA manipulations were almost exclusively done in a copy, cut, and paste manner. While this enzymatic approach has produced a wealth of scientific advances, implementing complicated biological designs with this method is the equivalent of writing a paper using a photocopier, scissors, and glue. Biologists have received their metaphorical typewriter.
Using their chip-based technology, synthetic biologists simultaneously synthesized and optimized all 21 genes encoding the 30S ribosomal subunit from E. coli.
Synthesis can be used to alter or improve sequences being built. In traditional cloning, targeted mutational changes are made only to a small amount of regions at once.
What do you do? The same thing as you, duh. I turn people's hair blue. That's because we're a blue gene.
One level of abstraction from the DNA synthesis is the parts production. Applying rational design to parts alteration or creation is advantageous in that it cannot only generate products with defined function, but it can also produce biological insights into how the design function comes about.
Directed evolution is a way around the design process. We use a library of DNA molecules subjected to shuffling and combinatorial synthesis. This allows us to accurately correlate the phenotype and genotype in order to enrich the molecule that produces the desired function.
Rational design and directed evolution should not be viewed as opposing methods, but as alternate ways to produce and optimize parts, each with their inherent strengths and weaknesses.
While rational design can make multiple changes or large-scale alterations that incorporate scientific knowledge, these strategies are rarely precise enough to finely tune a system's behavior. It's difficult to know if additional optimization is possible when employing rational design. That's why we use these methods in conjunction.
The cellular engineer of the future will not be restricted to the catalog of known biological parts, but will also have the tools needed to supplement natural parts with custom-made parts for specific applications.
The engineering process usually takes multiple cycles of design, optimization, and revision. Towards these ends, mathematical modeling is a useful design tool.
Various mathematical formulations can be used to model gene circuits. At the population level, gene circuits can be modeled using ordinary differential equations, also known as ODEs. Given the same initial condition and numerical configurations, different rounds of simulations will lead to exactly the same results. The ODE formulation ignores the randomness intrinsic to cellular processes and is convenient for filtering out system noise.
ODEs facilitate further sophisticated analyses, such as sensitivity and bifurcation. This is standard practice. These results would tell us how we need to design the circuit.
In a single cell, a gene circuit's dynamics often involve small numbers of interacting molecules. Such small numbers will result in highly noisy dynamics. For expression of a single gene, noise may be critical and needs to be considered. This can be done using stochastic models. Different rounds of simulation using a stochastic model will lead to different results each time.
It's a way to determine to what extent the design function can be manipulated and given a certain level of uncertainty. We can't accurately predict the noise, rather only account for it.
Mathematical models, either stochastic or deterministic, can be digitally evolved in silico to generate optimal circuit designs that satisfy a particular objective.
It's a rare and joyous occasion when a genetic synthetic circuit actually works for the first time. Because the more complicated the design is, the harder it is to put everything together.
Natural biological systems are astonishing production factories capable of synthesizing an impressive array of chemicals with relatively high yields.
Current methods for organic synthesis often rely upon exotic solvents. And they're expensive and they produce a lot of toxic waste.
If we can develop biological, catalyst, and synthetic metabolic pathways, we can usher in a new era of environmentally friendly chemistry.
As we break dependence on toxic solvents and catalysts, to decrease waste product formation, we can create green chemistry.
Total chemical synthesis is difficult and costly.
To engineer the yeast strain, we first had to increase precursor production, then manipulate the FPP pathway to augment its yield. Finally, we downregulated a gene that diverts FPP to sterol-producing pathway and added genes from A. annua. It was only to convert FPP to amorphidine and subsequently convert amorphidine to ateroministic acid. I'm glad I didn't have to pick up that check.
Drug production is an example of how synthetic biology can contribute to medicine. Billions of immunological cells patrol our bodies at any given time on the lookout for antigens that indicate foreign cells or abnormal function. A key feature of our immune system is that it is predominantly targeted to the particular offending pathogens or region of infection through the use of cell surface receptors and signaling molecules.
Sometimes drugs have unintended consequences. For example, chemotherapy targets to control the fast proliferating cells in cancer, but sometimes inadvertently damage hair follicles and gastric lining, so you lose your hair and your lunch.
We can engineer cells to target things in our body, cells in our body that aren't recognized by our immune system.
Cells have the advantage of being able to interpret and respond to complex environmental signals.
Tumor environments are often hypoxic and allow for high bacterial densities due to depressed immune function in the tumor. You don't want to know what that looks like.
Bacteria are easy to manipulate because their physiology is simpler than mammalian cells.
I think people would be more comfortable dealing with therapeutics derived from mammalian cells.
Recent parts design has produced a library of the so-called zinc finger nucleases, also known as ZFNs, that may enable in vivo human gene replacement.
The advantage of gene correction over gene insertion is that the replaced allele is present in its natural chromosomal locus. So there's a better chance they will be properly regulated, like magic.
As the global supply of fossil fuels diminishes, alternative and renewable energy sources will become more critical than ever before. Derived from crops, bioethanol has emerged as a potential way to convert abundant solar energy gathered by plants into easily stored fuel for combustion engines.
Production of bioethanol relies upon microorganisms such as yeast to ferment the plant materials. I wanted to call it carcohol with the slogan, "Why drive drunk when your car can do it for you?" Not a lot of people like that.
Bioethanol production is economically feasible only in the regions producing such crops. Synthetic biology could play a role by either boosting expression through system design or improving activity and stability through parts design. The human body is a complex system of specialized cells, tissues, and organs. Remarkably, each highly specialized cell in our bodies arises from a single fertilized egg cell. This process of differentiation and morphogenesis is mediated by the delicate interplay of chemical gradients, cellular receptors, differential gene expression, and cell migration.
Nature has produced incredibly complex systems, as well as a fantastic way of assembling them. Considering the intrinsic noisiness of the system, we begin to ask ourselves, in what ways can we recreate the complex pattern systems we find in nature? And how can we manipulate them?
Higher order functions in natural biological systems are associated with multicellularity and cellular specialization. To produce similarly complex functions, synthetic biologists will require mechanisms that produce and maintain differentiation patterns. These mechanisms may lead to highly sophisticated cellular systems for fabricating biomaterials with well-defined dimensions. This line of research may also synergize with research efforts focusing on regenerative medicine and tissue engineering, both of which hinge upon controlling differentiation and pattern formation. It has been suggested that many of the difficulties in the production and optimization of biological circuits are due to an improper and incomplete description of parts.
There are two things wrong. First, we're unsure of how some of the parts work. Second, even if they are known, there's no standard of measure. So, my transmission for my 67 Corvette won't fit in your 67 Corvette.
Imagine if we actually could standardize cell parts. I could be like, hey, my mitochondria are running low. Better get a new one.
If we can learn how to manipulate cells just by making new parts for them, the money will keep rolling in.
Given the amount of cell physiology that is still poorly understood, to what extent can we standardize parts or systems with confidence?
How much standardization can we afford and still hope to create useful systems that work in complex environments, like cancer or a polluted environment?
The true challenge lies at the functional levels. Parts will impact and be impacted by cell physiology, which also changes in response to the environmental conditions. Parts tested in isolation may unpredictably impact each other's functions when combined.
It's really hard to have complete confidence in a standard strain under standard conditions because we never really know how they're going to interact with each other, so that makes standardizing parts more complicated.
I like to use non-standard strains in my research. The stranger the better. The more diversity I use, the more I learn.
Standardization creates a combinatorial nightmare. The inevitable result is a standard for only a limited number of strains.
In addition to redesigning cellular processes, synthetic biology can contribute to producing artificial cells exhibiting all the qualities that we associate with life. It is probable that no matter what system is devised for artificial encapsulation of materials in membranes capable of self-reproduction, there will be argument as to whether life has truly been created. Somatic cell nuclear transfer, best known for cloning Dolly the sheep, has already accomplished a cellular cold boot.
The nucleus containing the DNA software is removed from the somatic cell, and it is no longer considered living by standard definitions and could be considered a collection of nucleic acid and protein molecules. An enucleated cell and cytoplasm hardware is not alive by consensus definitions. However, when the two are combined, life arises anew.
Where do we draw the line? Will it only officially be the creation of life if the new life is chemically synthesized from the old life? Will old life itself be produced from pure elements?
De novo cell design can actually shed light on both the properties needed to produce life and how terrestrial life could have arisen.
The field of synthetic biology is burgeoning.
It allows us to do things like use bacteria to fight cancer.
Programming on the cellular level so that they can perform specific tasks that will help, hopefully, the human body in the future.
We take life and we reorganize it in a way that's more efficient, and I think that's very profound.
While many of the advances are still years ahead of us, the call for research is immediate and relevant for the world in which we live.
Robots.
I want my very own Android so I don't have to do my laundry anymore.
At all levels of abstraction, parts, devices, and systems, mathematical biologists are modeling exciting new developments.
It shows that we are gaining more control over the universe that we live in. And if we can do that, we can do anything.
Whether the end goal is to create cancer-fighting bacteria, bioethanol-producing yeast, or genetically altered beings, one thing is clear, that science fiction will soon become science fact.
Just as the microsound... I'm sorry.
It's okay. Start over again. This will be in the outtakes.
Great.
So what can synthetic biology do for us?